I'm going to discuss Tuta absoluta and some similar moths this morning. My name is James Hayden. Again, I work for the Florida Department of Agriculture and Consumer Services and in a regulatory capacity. My office is inside of a University of Florida building inside the museum here. So the outline of this is the target, Tuta absoluta. I'm going to discuss a little bit about taxonomy of certain Lepidopteran families, in particular that of the target, the family which belongs to the family Gilligidae, and also the practice dissection subject, which is Helicoverpa, which belongs to the Noctuidae. I'm going to focus on just in those two families, but later Todd will get into discuss a few other families that might be of interest as pests. And finally, I'll touch on dissection methods, which Julieta will also discuss later. So first, we just went through the, what is a moth? We just went through this. A moth consists of, well, there's a head, thorax, abdomen, like all insects, a forewing and a hind wing. And the genitalia for dissection are inside the end of the abdomen, the posterior end. Just to be clear, I mean, there are some insects like, that dragonflies, for example, that might have a genitalia elsewhere on the body. Where spiders have them in the palpi. But like most insects, leps have genitalia inside the posterior end of the abdomen. Lepidoptera have a coiled tongue. This is one of the most characteristic things of leps, is that they have a haustellum or proboscis, which is coiled up, which has by which the adults imbibe fluids. They also have scales on the wings and the body, and 2 pairs of flight wings. The larvae caterpillars also have certain characters or features that we won't discuss in this training. So here are some photographs of other similar insects that I often get. They're often submitted when to me as things they think they're moths. But all of these things, none of these things have the characters we just discussed. For one thing, they do not have flat scales in the body. Some of them have hairs, which look similar, like this Trichopteran. But once you get them under the microscope, you can see that they're actually hairs. They're not flat. They're not spatulate. These things also have mouth parts that are different than. They're not a coiled tongue. They're not. The mouth parts might be for chewing. They might be for sucking, but they're not like a lengthy rolled up coiled thing. They have dipterans, that is to say flies, have only one pair of wings, not 2 pairs. The cycodid here has only 1 pair of wings. These other things have 2 pairs, but they don't have. We're talking about Herniptera, or there's one weird family of bark lice, Psocoptera. Sorry, that's misspelled. It should be Spsocoptera. That they're kind of hairy and narrow-winged, but you don't really have to worry too much about these things, because when you're looking on traps, most of the things that look like moths will be moths. So I'll go on to the next thing. Where are we in the big tree of life? To orient you all with Lepidoptera, this is one of the largest radiations of insects, with about 160,000 described species. And here's a big phylogeny of it. The Tuta absoluta belongs to the family Gilligidae, which is in this red circle here. Family Gilligidae, superfamily Gilligioidea. To orient you, the butterflies are just over here in this group, and the Noctuoidea with the target dissection subject, Helicoverpa, is down here. So we're going to successively kind of zoom in on this. The superfamily Gilligioidea includes about roughly 21 families and 16,000 described species. They're global in distribution. They're all kind of little moths that look like, this is a plate from the Moths of America, North America, of Mexico, fascicle. Just to give you a taste of what these look like. Galechioids have a few distinctive characters. For one thing, they have the haustellum, or the tongue, has scales on it. It's scaled, unlike the naked haustellum you saw earlier in the other noctuid. This here does not have scales, whereas you can see scales on this tongue. Can you all see this?
Yes.
Can you see this? Okay. I'm not sure. My little Zoom box thing is covering some of the screen.
It looks really good.
Okay. Okay. Now, to be clear, there's one other big radiation called the Pyrroloidea that has the superfamily Pyrroloidea also has scales in the tongue, but most pyrroloids do not have palps that go up like this. These are mouth parts, the labial palps that stick up in an arc and come to a pointy tip end. So when you see this combination of two characters, it's usually a Galechioid. The head also tends to be smooth scaled. They look like, well, they look like my hair right now. Now at 5 o'clock today, my hair is going to be stuck up like this because I've been pulling it out all day. And then it will look like a Taeniid or a Bucculatricid or a member of a different superfamily. This is another, whether the hairs kind of look like they're disheveled or not on the top of the head is actually a good character for many micromoths. So when we say a smooth scaled head, is the hairs are neatly combed down like that. Okay, so the family Galechiidae is one of those 21 families in the Galechioidea. There are about it's one of the largest ones. There's about 500 genera, but nearly 5,000 species that are described. It has the characters I just mentioned, plus a few other characters. One of them, some of these are genitalic characters, which I will not discuss in detail. An easy diagnostic character for the family, though, is that the hind wing comes out to this pointy tip right here. It's generally a good character, by and large. And some things don't have it, and there's a few things in other families that do have it. But it's, by and large, Galechioids tend to have a pointy tip and a hind wing like that. They tend to be kind of small. Now, I'm going to make you give you some assurance here. The higher level families in the Galechioidea are often in flux. There's a lot of confusion out there. Things, you know, one family now is a subfamily or a tribe and so forth. But the Galechiidae as a family tends to be pretty stable. Okay. So now we're going to focus in even further. Tuta absoluta belongs to this tribe called the Gnorimoschemini. Now, there are several tribes and subfamilies within the Galechiidae which are also in flux. Again, the same kind of confusion that I spoke about with the families in the Galechioidea also applies to the subtaxa within Galechiidae. The Gnorimoschemini tends to be pretty stable, though. It's been received a lot of treatment over the years. They're really hard to identify, though. So I'm telling you this because you'll see this name out there, the tribe the Gnorimoschemini. Don't really fret about being able to identify a Gnorimoschemine. They're identified by, diagnosed mainly by female genitalic characters like this hooked signum and these little lateral microtrichia around the ostium of the female genitalia. There's about 900 species, 400 genera, diverse in the neotropics. This group was mainly dominated by a singular, the taxonomy was dominated by this Czech worker named Dalibor Pavalny, who passed away in 2004, who produced a prodigious amount of papers. I have a thick file about 4 inches thick of Xeroxes of things that he produced over about 4 decades of cranking out species descriptions and revisions. Since he passed away, other people have filled in the gap and tried to revise and improve his work and make things more accessible. he was the first one to transfer Tuta absoluta into Tuta as a genus. The normal schemines are important because a lot of the pests of tomatoes and Solanaceae fall in this tribe. They belong in this tribe. This includes Tuta, Kypharia, Thoramea, Symmetroschema, species of Tecia. There's even a few species that are pests that are of other families, such as Urosacca. Many of these things are covered in this online website, the Microlepidoptera on Solanaceae. You can go to. I and collaborators produced this website a few years ago in 2013. So the hosts in this group, host plants in this group are, sorry to make you jump around, various families, including Chenopodiaceae, Asteraceae, Caryophyllaceae. But like I said, most of the pests on Solanaceae are concentrated in this tribe. The top three photographs are photographs of really bad damage on Solanaceae, on tomatoes from greenhouses, thanks to the EPPO website. Down here, we have a little picture of Kypharia in tomato. This is the native tomato pinworm that's native in Florida. So far, all the damage that we see on tomatoes in Florida is this species. But this is what we're watching out for, what we do not want to see show up in Florida, up at the top. Down here, I'm sharing this one photograph of something in Atriplex. Just for your reference, Atriplex patulin amaranthaceae is a different family, but I'm going to mention this weird little critter later on. So to be clear, most of these species of moths feed on only one family. So if it's Tuta absoluta, I am not aware of there might be some records of it feeding on crops that are not Solanaceae, but they're really peripheral. They're not much to worry about. Here's some other fun stuff, interesting things. The genus Symmetroschema, for example, another Gnormoschemine genus. These things feed in things like peppers and physalis. Some of you, since you're all in the Caribbean, have any of you heard of the pepper bud moth, Symmetroschema capsicum?
Yes, yes.
Yep. You encounter it once in a while?
Not much these days. Previously, yes, but not a lot these days, no. But one thing that you mentioned that I'd like to just add a little bit more about is when you get down to the taxonomies, these things, sometimes the taxonomists change the names of these insects, the genus as well as the species, and you think sometimes that you're dealing with something completely new or different, and unless you go and actually look it up to see if it may be a synonym of something else, you might think you're dealing with something different altogether. So I said to myself very often, why can't these bloody people make their minds up?
I know. I feel the same way. I am both a producer of this confusion and a consumer, because my specialty is in the Pyraloidea. That's what I got my training in. So I'm always changing the names and in what I publish, but at the same time, my job is to identify everything, and I easily get confused when my colleagues change the names of things. This is Tuta absoluta itself has been placed in, I don't know, 4 or 5 or 6 different species, genera over the years. It was originally described as Thoramea, then it was placed in Scrobopalpula, and then Scrobiopalpuloides. It's been bounced around, Normoschema itself for a while. So we're calling it Tuta for now. I'll explain it a bit. I wanted to just mention this one other thing over here. Symmetroschema, there's an undescribed species of Symmetroschema in this wild plant called Lycium carolinianum. This is a cool thing. I know I've caught these things and raised them before. There's a lot of new discoveries out there. Tuta itself was described by Merrick in 1917 in Thoramea. Native to Peru, it was bounced around, placed in one genus after another, and transferred to Tuta by Pavolny in 1994. Yes, that might change in the future because it doesn't really fit very well in any genus. But we're calling it Tuta for now. There are some Western species that have been placed in Scrobopalpuloides, Pavolny. The type species, if you're familiar with the concept of a type species of a genus, the type species of Tuta is something called Tuta atroplacella, which is described from Argentina, and it feeds in a species of Atroplex. So I kind of suspect that there's more additional reason to think that absoluta might belong elsewhere, but I'm not going to go there right now. What matters really is the genitalia. So on the left-hand side, we have the male genitalia, and the female is on the right, just for comparison, for your reference. Normoschemines are really, really boring brown moths. Many of these things have almost no wing pattern at all. Look at these things. Yeah, there's hardly any pattern. So, yeah, the genitalia show usually consistent differences that are discrete and consistent differences. These are the basic parts of a moth genitalia. These clasping things are called valvae. This whole object down here is called the vinculum. This up here is called the uncus, and this little thing is called the nathos. I'll show these parts in on. Here's a scan of some of the tomato pests in Europe. The Normoschemon in Europe, a fantastic book by Peter Humer. Down here, there's the potato. Could you please mute? Somebody has their phone on. Could you please try muting yourselves, y'all? Thank you. Good. So this is in Europe, there are three potato pests. There's the potato tuberworm, Thoramea operculella here, Tuta absoluta, and Kypharia lycoprosicella is this one here. And you can see these interesting things at the end of the valvae. Here's the uncus of Kypharia, a long pointy thing. These are illustrated much better on this website, Microlepidoptera of Solanaceae, that we produced. These shows a number of target taxa. So now I'm going to go on to the pheromones. I expect all of you will be running pheromone traps, and this really helps because only a limited number of things are attracted to the same pheromone as Tuta absoluta. We'll find out what other species might be attracted to it, but it's not much. Our experience in Florida is a few things. And Julieta, I'm going to ask you to please chime in and correct me where I'm wrong, because really you see a lot more of the traps than I do. Probably all of them, the trap catch. I think that Kypharia lycoprosicella is also attracted to Tuta pheromone.
It is the main thing we see.
Okay. Mm-hmm. And the species of Sinoe, Capsana, was an interesting discovery. A new species that was described based on trap catch.
Yes, we're actually finding this genus in Tuta traps in other countries as well. Mm-hmm. So we know that in general, this genus is attracted to the Tuta absoluta lure. Mm. So everyone should expect Sinoe in their traps.
Mm-hmm. Sinoe itself is not a member of the tribe Normoschemini. It's belongs to a different tribe. The genitalia are a little bit different. This is the uncus here, but nevertheless, it's a little gray moth that you can, that turns up in traps. Several years ago, we got one specimen of something from Panama City, and I mean the Panama City, Florida, not Panama City, Panama. This is out in the along the Gulf Coast. And we visited the site, and it turned out, it looks like it was a species, possibly an undescribed species related to the Western North American Tuta species. We got one turned up in a trap. It was preliminarily identified as something related to a Western Chiquitella, which feeds in Atroplex. This was a little interesting detective case. We went to the site. We drove it. It took a day to drive there. Florida's a big state. And yes, there's actually Atroplex on the shore. We found leaf mines and successfully reared out one specimen, but it was a female. And it didn't look exactly like what we were expecting. It looked like a different genus, I think. But nevertheless, there's something there. It's really cool for us because it's Western radiations of moths that are diverse in Western, like in California, sometimes have vicariant representatives in Western Florida. And this might be such a case. But it's hard to get out there because nobody has time and money to get out there. And I went out there on a couple of weird, getting out there means, sitting in the car for 5 hours, and, I'm sorry, I just don't have the time. And one time I did go back there a couple of years later, the Atroplex were washed away by a storm. So if somebody could just get out to, the Florida Panhandle and pick some leaf mines, they would probably find new species out there. But this caused some alarm bells because it turned up in a Tuta trap in a little, Julieta, I think it was in a little garden. It was in a community garden next to the shore, right?
That's correct. It was an organic garden from a community.
Mm-hmm.
A single specimen in a glue trap, so we didn't have much to go on except that one single specimen.
Mm-hmm. Well, fun discoveries. This is what a glue trap looks like. And traps can also get covered with other kinds of random insects. So if you see a whole bunch of moths on a trap, then these are flies of some kind, but if you see a whole bunch of moths, they might actually just be feeding on detritus or other stuff. In Florida, we often see a lot of tineids just on traps, only because they occur in big numbers, feeding on garbage or whatever in their environment. For example, the so-called dancing moth feeds on fungi. Zistrologa, one of these species, Zistrologa grenadella, this is described from, I think, the Virgin Islands. I forget where, another Caribbean island. These things are very abundant in South Florida, where they eat bark mulch. Another species that turns up on traps often is what I call the tribble moth, Acrolophus walsinghami, native to Puerto Rico, got into Florida about 20 years ago. Yeah, this thing is probably the most common urban moth in any city from Tampa southward. But we don't even know what they're feeding on. They're probably feeding on grunge and grass thatch. But you'll find traps covered with them. So does anybody have any questions before? I'm going to turn now to dissection methods. Does anybody have any questions, point or comments? Was I erroneous about anything?
Hi, Jim. I just wanted to comment really quickly. If participants aren't comfortable just speaking out, you can place your question in the chat to everyone, or you can raise your hand and be recognized if you prefer to be acknowledged in that way.
So I'm going to turn to the dissection subject now. This is Helicoverpa armigera. This is what we'll be focusing on for the dissection. It belongs to the family Noctuidae, which is an even larger radiation of moths, Noctuidae sensu stricto. Well, it depends on how you carve it up. The haustellum or tongue does not have scales on it. They're usually much larger than Gillikiids, and the hind wings are broader. They don't have this apical, they're kind of a D-shaped hind wing. They don't have the apex produced to a point. A character that you might be interested in is that they have a pair of ears, tympanal organs on the metathorax. If you get a good photograph of them or a good look at one, you'll see this hole in the back of the body, just on the anterior end of the abdomen. It's kind of in the same, roughly in the same position as that grasshopper that we did in the warm-up exercise. A little lesson about how to sex moths. If you are in doubt about how to, whether you even have a male or a female, this is the wing coupling mechanism. It's pretty reliable for to tell a male from a female. Male moths have this, this is the anterior end of the hind wing, and this is the anterior end of the forewing, and they kind of fold up closely to each other. The hind wing has this one or more frenular bristle that sticks out in front, and something in front that clasps it, that holds it in place. In males, it's the extension of the radial or the subcostal vein of the forewing. I mean, it's actually a membranous thing that sticks out and holds a single bristle. Whereas in female moths, they only have a bunch of scales or aceti coming off of the one of the veins, and they have multiple bristles, at least two. Okay, there are exceptions to this. There are a few taxa where the females have only one bristle, like the males. Sometimes in some things this is lost, but for 90% of moths, this is the way to sex them. Here's some photographs. Males have a single bristle and this little thing that comes off. It's covered with scales, but if you were to descale it, you'd see this little thing that kind of wraps out and comes out and grabs the bristle. Whereas females have two, and the retinacular hook is not present. They're only held in place only by these a bunch of scales. Now, half the time you can see the genitalia themselves directly, but when their genitalia are retracted into the abdomen and you can't see them, this is what you do for telling whether you have a male or a female. Now here's some parts for generalized male genitalia of Lepidoptera. I'm going to keep focusing on the male genitalia because this is what you should expect to see in the traps, because the males are attracted to the the pheromone. Again, these two clasping things in the sides, these two sort of wings, are called the valvae. Up at the top, the dorsal side, we have the uncus, which is a thing that kind of comes down like that. Sometimes usually it's a long thing that sticks up, but in some things like Tuta, it's a very reduced and kind of, well, you'll see. There's something called the gnathos down here. The gnathos is not present in noctuids. Here's the uncus, gnathos, valva. And then the phallus, which is also called the adiagus, is the intermittent organ. And it comes through here, and it's removed in this photograph, but in this illustration, but it comes out here. Here's the noctuid, which you'll be dissecting. Here's the valvae on the left and right, and the uncus coming down from the top. And the phallus is this thing, it's always removed in the dissection. The phallus would be coming out of here, through here, but I've taken it out, and this is what it looks like. The vesica is this sac inside that pops out under pressure. It can be removed. It's very difficult to remove in Helicoverpa because it's so long and it's covered with these spines that are called cornudi, kind of jammed up inside, but it's partly everted in this dissection. And the females are just for your reference, just for your comparison over here. Again, I don't expect you to see them in traps, but you might see a female if you were to raise it from a host or to catch it at light. Raise it from a caterpillar. This thing is called the corpus bursae. This is where the spermatozoa are stored until fertilization. And there's also an appendix bursae, which is really enormous in Helicoverpa, but this Chinia species down here is related, and it shows a rather more normally proportioned sizes. The ovipositor is where the eggs come out, over here. Gillichidae have some special dissection methods that are really, I'm jumping into really advanced tips right now, so you can go get it, you can ignore this slide if you want. But for one thing, don't be surprised if Gillichids take a little bit longer to cook in KOH than most moths. Some people say 30 to 40 minutes. I don't really have a problem with more than 20 minutes. In the illustrations, you'll see it's good to unroll the genitalia by severing the vinculum right here. So this is, it's kind of hard to explain, but this end corner right here is usually attached to this thing over here in most moths. And orange G is a useful stain for Gillichids. I mentioned this second point, unrolling the genitalia, because remember that guy Pavolny, the taxonomist who produced tons and tons of publications over the years? He really dominated the taxonomy of the Gnorma schimini for several decades. All of his illustrations look like this. Yeah, they're really confusing because the parts are kind of folded up together. And these really interesting diagnostic things that stick out, the subbasal parts and the sacculus, are kind of, they're on top of each other and overlapping. These are the valves on the sides. It's not easy to see what he was thinking. And so all the modern workers, that in the stuff you'll see online, things are kind of folded open like a book, okay? You've got this, instead of being folded up like this, what they've done is they've opened it out like that. You don't really have to do this. I don't expect. I think that in your diagnostic dissections, you'll be able to see Tuta absoluta, even if the genitalia are not properly opened out like that. But just to explain where things are. Does anybody have any quick questions? I'm going to go into a few last slides. I know I'm probably running over time. How am I doing on time, Dr. Hodges?
Dr. Hayden, you're just fine on time. Why don't you just keep going over a few more things? I think this is really important content. I want to remind everyone that you can place your questions in the chat to everyone, or you can also raise your hand and be recognized by Dr. Hayden if you're more comfortable with that.
Okay, thanks. I'm not sure where the chat button actually is. I'm screen sharing now, so I'm not, am I missing anybody's chat questions?
I don't think so, because I have my chat open, and so yeah, it's the chat button should be on your toolbar section of your screen.
Okay. I'm not sure if I can see it really. Yeah, could you please just interrupt me if you have any questions in this last bit here?
Yeah, I have a question.
Yes, yes.
Would you mind just going over quickly what you were talking about when you were looking at the wings, how to differentiate the male from the female with the hook and all that?
Mm-hmm. Yes. So do you want me to review this and repeat it?
If you don't mind, quickly.
Okay. Yep. So in males, I'll show this photograph first. In males,
well, that's the one I was trying to figure out there.
Yeah. So they differ in males and females differ in two ways, two aspects. One is that males have a single bristle and females have more than one, two or more. These bristles come off of the anterior corner of the hind wing down here. It's obscure under scales and hairs, but they emerge from the hind wing. See? This is the hind wing here, and these bristles come out of it in the upper corner of the hind wing. The other aspect is that males have this little membranous hook that comes out of the subcostal vein and wraps around and holds, clasps the frenulum. It's called the retinaculum because it retains these bristles. It hugs them. But in females don't have this little membranous thing. They only have a bunch of bristles or scales that hold the bristles in place. These things here. So these are equally effective. They holding the wings together. This is how the wings are kind of held together as the moth flies. Otherwise, they'd be beating independently or get out of sync. So does that make sense?
Yeah, thanks a lot.
Okay. Now, granted, some things don't have this. For example, butterflies have lost all of these mechanisms. The butterflies have really big, broad wings, and they're able to keep the wings in sync just by friction on the wings. And there are a few moths where these are, it's different. But for, I'd say, 80 or 90% of moths out there, including the targets that we're looking for, this is how to reliably sex them. Tell the difference. So I'm going to go to review dissection tools here. Dissection is pretty simple, and it's actually, we've been doing moth dissections for, since people really started getting into them in the 1890s, so about 130 years. People knew about butterfly genitalia even earlier than that, but moth genitalia dissection is kind of a cult thing. Some people really get into it. I'm one. Dr. Gilligan is another. Julieta is another. Some of us do this at home recreationally. Yeah, in fact, a lot of the stuff you'll see online is done by a few dedicated amateurs. The tools are very simple. You basically need something to dissolve the fats and the greases of a moth abdomen. And that is potassium hydroxide or sodium hydroxide. Usually potassium hydroxide, but lye, sodium hydroxide, I've found that it's a workable substitute if you can't get KOH. So these are the parts that I use for dissection, the things that I use. Basically, when I say snail juice, that's on my desk. This is simply tap water with a drop of detergent. With soap in it, like dishwashing soap. That's water with just a little soap to break the surface tension. So you have a fatty, greasy abdomen. You've gotten dissolved most of the fats, but in the KOH, but it's still floating around. This is what I dissect in. What I prefer to dissect in is slightly soapy water. And it's what I do is I fill a vial with 10% potassium hydroxide solution and put it into a double water bath. So these are vials full with the KOH in it. I put an abdomen into here and then let it cook at 100 Celsius for, simmering, boiling for 20 minutes. And then I take it out and I put it into the water. Oops. We'll go through this again. I don't have any step slides, but this method has been published about in numerous places. Probably the best place for this is resources here are, I'm going to jump around a bit. Todd Gilligan's Tortricids of Agricultural Importance. This is probably the best single web page for well, really everything. He links a lot of dissection resources through one of the pages on this. Yeah, this is it right here. ID tools, LEPS, Tort AI, dissections. This has a ton of references, PDFs that repeat this basic method for dissection. So it's not hard to find. For storage, after you've dissected the genitalia, putting things into a micro capsule a micro vial full of glycerin is what is good enough. It might be kind of difficult to fish out the genitalia, but if in either case, for really small things like Tuta, but it allows it to be kept with the specimen on the pin for future reference. Don't forget to kind of burp it like by sticking a pin in and then pulling it out to remove any air. Otherwise, the cork will come out. It's just like an alcohol vial. When you stick the cork in, you stick the stopper in, make sure that you remove the air so it's not going to come out 5 minutes later when you turn your back and put it in the drawer. I'm also going to mention, these are the steps for slide mounting in Uparol. Probably only a few of you will do this, but if you're going to be producing a reference collection of slides, this will be really good. Leave genitalia for several hours in ethanol, transfer to 100% ethanol, transfer to Uparol essence for 5 minutes. Meanwhile, clean a microscope slide. I'll just go through this visually. After you've made your dissection, you put in 100% ethanol or 95% ethanol for a few hours to remove any water. Then you can optionally place it into 100% ethanol to remove any residual water. I prefer to mount in something called Uparol, but you can also use the balsam. Thank you. Uparol essence for a few minutes. Prepare your slide. Then you blob a drop of Uparol mounting medium onto the slide here. You put your genitalia onto that, and then you need a little bit more of the essence just to make things go down without any bubbles, and you put a cover slip on top of that. And then you wait some period of time for the slides to set. Wet Uparol might actually take days or even weeks to set properly, depending upon your dissection. If it's a big fat noctuid like the Helicoverpa, it might take a couple of weeks. But with Tuta, a tiny little Gilligan, days would be fine. And a really important thing is to label your slides. Don't let slides accumulate without labels. Here's some more, as I said, some more dissection resources. Genitalia images they're all out there. There's some interesting websites that you might want to click on, like the UK Lepidoptera Dissection Group, Moth Photographers Group. Most of these things they're kind of hit or miss. I mean, it would be great if we had like all genitalia online, like the UK Lepid Dissection Group. That'll happen probably in the 23rd century when the rest of the world catches up. But for certain target pests like tortricids or Tuta, we do have genitalia online. For those of you in the Antilles, here's a fun website in French. There's also an English version that shows a few noctuids down online. That might be kind of golly gee whiz fun, but this is more of a kind of a faunistic thing, not the agricultural, not the regulatory. And of course, don't forget, many publications have been put online through the Biodiversity Heritage Library or just scans of this and that. So if you're looking for a scan, just Google it. And you'll more likely than not find out a publication with that's online. For curation, I'm going to back up a little bit. This is how I prefer to dissect and, to pin things. Pinning either with a micro block or a double mount like this. You can also put things in gel capsules. I expect that a lot of you will use the gel capsules because first of all, the moths that you pull off of this, if you get a target, if you get Tuta absoluta on a trap, you will take it off the trap and you've got to keep it somehow. Don't throw it out because it's then, we're all regulatory agents. We need to keep the proof that this pest was actually here. You got to keep the body. But it's probably going to be delicate. It's going to be, many of you might have good pinning skills. It's going to be fragmented. So put it into one of these gel capsules and then stick that onto a pin. Thereby, you have kept the body. Other people can take it out of that and examine it. There, it's good. Another good point is it might be better for DNA sequencing if you don't go through a relaxing process. And Todd can speak more about that. What's good, what's bad, what works, what doesn't for as far as DNA goes. I'm going to show you, here's an example on the left-hand side of why we keep the bodies, why you keep the specimens. Pectinophora gossypiella is a major pest of cotton in the New World. And it has been successfully eradicated from all cotton growing regions of the United States after a decades-long, many years-long, costly process. Last 1 or 2 years ago, U.S. Department of Agriculture finally declared success after waging this war, except that it is still present in wild cotton and a few sites in southern Florida. These are far away from cotton-producing areas. So when we told USDA, a few years ago, Florida Department of Agriculture, had to remind the USDA that, yeah, it's still present in South Florida. And they initially questioned us and they said, well, are you sure? And we said, yeah, here's the specimens. We'll lend some to you because we kept them. We didn't throw them out. Here they are. Here's the proof. And so the USDA had to go back and kind of revise their assessment and, okay, say, back to the drawing board. We're going to go through this one time again. And, it was a few years later until they actually declared it successful. See, this is why it matters to keep the specimen if you think you have a target test, because other agencies, other people might question you about this. This is science. We keep the evidence. And just to go along with that, data. I know that everybody's so busy. And we often write only a collection code and stick that onto the specimen. And then that code gets lost in it's written down in a notebook or a hard drive on an Excel sheet or something. And then it gets misplaced and lost and thrown out. So I can't emphasize enough. Please get the full data onto the specimen, on the pin, as soon as possible after you collect it. Because people come and go. I might get hit by a bus tomorrow and, or something might happen to me. And who knows where my notebooks are, and who knows who can recover this stuff on my hard drive. It's really important to label your specimens properly. This is the kind of order that I prefer, going from the highest to the lowest geographic coordinates first. USA, state, county, city, geographic coordinates, and then I get into like the details of the trapping method. Here's the date, the collector, and this little code number here refers to the database number in our regulatory database. And I like to make small labels, print them really small enough so that like this, so that it's appropriate for they can actually fit them into the unit tray. And that's everything. I've come to the end of this presentation. Thanks. Does anybody have any questions or comments?
Thanks, Dr. Hayden. That was really quite excellent. We really appreciate your time today with this. Any questions before we go to our breakout? We have some claps, so thank you. Everybody seems to have really appreciated the presentation. So thank you, Dr. Hayden. Yes, the virtual clap, and we really appreciate your time. We're now going to go into our breakouts where we are going to, we're actually going to focus on thinking about Lepidoptera morphology. Excuse me, we're actually going to focus on, in the breakout, we're going to focus on this Tuta absoluta identification. And so that's going to be the focus of this next activity. And we're actually going to have an extended breakout time. So we're going to return to the main room at 10:45. So plan to return to the main room at 10:45. So if your group takes the entire time, that's your choice, but I would suggest that you try to end a little bit before 10:45, just to give everyone a little bit of a break before returning to our next presentation, which will be the preparation of specimens for dissection using male corn earworm. It's going to be Julieta Brambila. And we're actually going to need you to have your supplies out with the KOH, because you're going to start thinking about how you're going to break your Helicoverpa abdomens and begin soaking them for tomorrow's activity. So we're going to go ahead and enter the breakouts now. Any questions before we enter the breakout?
Yes, I have one question. Were we supposed to receive sample specimens?
Yes, that would have been in the sample from the University of Florida.
Okay, because I only received the books and the pocket microscope.
Okay, so Sarah, maybe you can talk to, what country are you from?
St. Lucia.
So Sarah will talk to you about that.
Okay.
Received a package with specimens from the University of Florida. So we'll talk more about that when we come all back together as well.
All right.
For letting me know that.
Thank you.
How's everybody doing?
We had good questions, and we even had some slides, some cleared specimens from KOH.
Oh, from one of the participants?
Yes.
Wonderful.
Yes.
Already getting started. So we're going to have to really, I guess, try to keep on schedule now to finish out by 12. And so, Julieta, are you ready for your presentation?
I'm ready. It's a very short presentation. Okay. I hope you have received your specimens for Helicoverpa. We're going to be practicing tomorrow dissection of first large specimens before we try small specimens. An idea we have is that if you do not have specimens of Helicoverpa, you can use any moth specimen that you have in your collection leftovers, or collect some at night near the lights of your house, so you will have some specimens to work with. If you do not have any specimens, don't worry, you'll still learn a lot looking at our pictures. Of course, we would like you to have hands-on practice, but do not worry if you don't have any specimens. This is a short version of the presentation tomorrow. I learned that your specimens were shipped to you in alcohol, so there'll be a minor difference in this training. At the beginning, it is the same. The main step is to remove the abdomen from several of your moths. The abdomen breaks naturally at the connection with the thorax. You just push it up, and usually that breaks the abdomen rather than cutting it or pulling on it. Try not to break them in the middle or at the tip. So just push on the abdomen upward and it will probably just separate. So do that to several specimens today, even while we're in the other talks, because the longer you treat the specimens with KOH, the more likely you will have a cleared specimen tomorrow. If your specimens were dry, you would soak them in alcohol for a little bit. If your specimens are already wet, damp from alcohol, you can skip this step, but make sure these specimens are in alcohol before the next step. So add the alcohol, soak for 1 to 2 minutes, and then remove the alcohol. On the next step, you're ready to add 10% KOH. I assume you already have your solution. If you only have pellets, you can make a solution, but make it a few minutes, like 15 minutes before you add it to the vial so that the pellets dissolve. After you add the KOH, make sure you cap your vial so that there is no accidental spill of the KOH anywhere. Wear gloves if you can, because it's a caustic fluid. It can irritate your skin. Leave them overnight for 24 hours, if possible, from today to tomorrow. But if you can find a warm place in your house where you can put it, it will improve the clearing. You can put it near your kitchen, but not if you have children. So be careful where you leave it. But a warm place is better than cold. And we hope that after 24 hours, we're ready for dissection. It's easy, just add KOH, wait overnight. The fluid will look yellowish, as you see on the left. And you're ready. The step on the right is for tomorrow. Tomorrow we'll start dissection. So that's it. That's all you have to do. Soak them overnight for 24 hours if you can, in 10% KOH. Now I'm ready for questions.
You could go to the slide, please. I kind of missed a step.
1 second. I will go. This one?
Yes, step three. Yes, okay.
After you break the abdomen, soak the abdomen in alcohol for a couple minutes. Then you remove the alcohol. And then you can add the KOH.
Okay, thank you.
All right. So we do have some questions in the chat, and so let's go over those. It looks like, Julieta, you already went back to step 3 and went over that, so that's good. And then Annette was asking, the specimens, are they the ones that we received in alcohol? So yes, you'll be using the specimens that you received in alcohol. The way we received these specimens, we just thought it was best to ship them in alcohol. So that's, a little bit different, but I think Julieta has covered that procedure. Are there additional questions? With that, I think we are ready to move on to Dr. Todd Gilligan's presentation. Todd, are you ready to talk to us about other families of pest significance?
Yeah, let me get the presentation started here. How's that look?
Looks great. Thank you so much.
Okay, good. So I'm going to talk to you about just some other families of Lepidoptera that you might encounter when doing any sort of survey. So the pheromones that we use to survey for moths are sometimes very species specific, and that's the case with Tuta for the most part, but you're likely to encounter other moths in doing any sort of survey, and so it's useful to just see some of the diversity that is out there so that you can recognize if you're looking at the target or some other moth that you might not have thought you might encounter. And so I don't expect that you would, of course, memorize everything I'm going to talk to you about today, because we're going to go through it sort of quickly, but you'll have these slides to reference in the future, and just looking at different pictures of moths can help greatly in figuring out if you're looking at the target group or some other non-target that you can pretty much ignore if you're doing a specific pest survey. So within Lepidoptera, there's about 157,000 described species, so that's a lot, and 124 families. This is the largest lineage of plant-feeding organisms, and so there's a lot of species that you might encounter in doing any sort of survey. So I just had a question here down at the bottom, how I can tell if this is a moth. And just so going back to what we learned this morning, 2 sets of wings, forewings and hind wings, and this coiled tongue or proboscis that's used for feeding, and then of course scales on the wings. And those are really the 3 main characters that we can use to figure out if something is a moth or not. So there's a lot of families of Lepidoptera, and luckily the major pests or other species that you encounter are only in a few of these families. There's a lot of families with small moths in them that are very obscure that you normally would not encounter. And so we're going to talk about these different superfamilies I have listed here. And at the very end of the presentation, the 3 families, superfamilies with the most number of pests we'll talk about at the very end, and those are the ones that you might be most likely to encounter. So just starting right off here with the first superfamily, Teneoidea. This is a superfamily with 3 families in it, a lot of described species, about 3,700, but it includes some very common moths that you might be familiar with or have heard of or seen before. And that includes clothes moths and bagworms. And so the Teneids in general, you can see the 2 pictures at the top here, are the group that contain the clothes moths. These are tiny, for the most part, very small moths. Most of them have some sort of spiky scales on the head. It looks like their hair is standing up or something along those lines. Also, sometimes the forewings are sort of shiny, as you can see in this specimen in the upper right. And if you've ever heard of moths eating clothes, eating sweaters or something like that, and people refer to clothes moths, those are generally in this family of Teneidae. A lot of the other species feed on dead skin, or you can even find them in decomposing bodies and things like that. A subfamily of Teneidae, and Jim talked about this earlier this morning, are the acrolophines now. They're a subfamily. These moths have these really long palpi that come up over the head. They look like antlers that come up over their head they're pointed back towards the body. And so that makes them really distinctive, and there's some very common species of these that are encountered pretty much everywhere, including in the Caribbean. And so it's just another group that you might encounter commonly. They're all brown, and they look sort of fuzzy because of these long palpi that go up over the head. Their head looks sort of fuzzy. Bagworms, the ones at the bottom here, make these distinctive bags on trees usually. You can also find them on houses or structures or a variety of places like that. And they live within these bags. The adults you would rarely see because you would normally find the bags, but the adults look like these examples here on the left. And the larvae live in these cases, and a lot of the females as well live in these cases and don't actually ever emerge from the case. They reproduce in those as well. And so these are just some common examples that you might find within the Teneoidea. The gracilarids is another group of moths that are fairly common. There's not as many species within this group, but these are leaf or grass miners. And so they are very small. The larvae mine within, like it says here, leaves or within grass. They make these tunnels that are somewhat distinctive. And so as a consequence, the adults are very small. These are generally less than 1 centimeter across. They have some characteristics here that you can use to identify them. But generally, when you see an adult, it has this sort of distinctive resting posture with its legs up like this and its antennae across the body and sort of in this triangle tent-like position here. Generally, there are some pests in this group. This middle species here is an important pest, but or related to an important pest, but these are not something that you would generally encounter in large numbers in doing a pest survey. These just might be consequential things that you would find in a sticky trap, for instance. The Eponomutoids is another large group. There's 10 families within this superfamily. I won't talk about the really specific characters here, but just show you some examples. So the Eponomutids are usually distinctive in that they have white wings, a lot of times with black spots on them, like the one in the upper right here. You can see the wings are quite rounded in this example here too. That's another characteristic of this family. The Ativids are webworms, and these are common in a lot of areas. They have this distinctive sort of checker spot pattern here with the orange and yellow and black, and that's really distinctive of a lot of species in that group. And then the plutelids contain, this family contains one of the most common pests in the entire world, and that's Plutella xylostella, the diamondback moth. It is a pest of brassica or cabbage that's found worldwide, and it's found everywhere, really very, very commonly. You can tell these moths by their small size. Plutella xylostella has this really distinctive wing pattern with this white along the dorsal margin and then brown in the rest of the wing. If you look at the head of these things closely, you'll see that the labiopalpi are kind of feathery here at the bottom, but then they have this segment that projects up here, up towards the rest of the head. And that's sort of distinctive for this group. And so Plutella xylostella is something you might encounter in any pest survey pretty much anywhere in the world. And then the Glycioidea is the superfamily that contains the Gillikids, of course, and Jim's already talked quite a bit about this. They have a scaled proboscis, and they have these long, usually slender labial palpi that are curved up over the head, and that's really distinctive of this group. A couple subfamilies, well, a family within the Glycioidea is the Elachistids, and then a couple subfamilies within that family are the Ethmians and the Stenomatines. The Ethmiids all sort of have this similar type of pattern. These are found commonly in various parts of the world, usually in the Western, certainly Western North America, they're very common. The group that you might be more interested in is the Stenomatines, and that's this group down here, the 4 pictures on the bottom right. This contains the very common or very important avocado pest, Stenoma catenifer. And this is a very important, probably the most important pest of avocados in the world. It's found throughout the tropics here in the New World. And so that's something that you might encounter in areas where avocados are grown. They're not too distinctive. The moths are sort of just kind of brown with some spots on them. But it's a very diverse group, and there's a lot of undescribed species. And if you look at the DNA of just Stenoma itself, there's potentially between 200 and 300 species of undescribed related moths to this Stenoma catenifer. So it's a very diverse group in the tropics, and a group that's not very well studied at this point. Echophorids is another group within the Glycioidea. These have some distinctive patterned moths, nothing that you would normally really encounter during pest surveys, but just an example here, you can see the curved labial palpi that are coming up over the head. That's really distinctive of Glycioids in general. The Coleophorids are a group within this superfamily that are case bearers, and so the larvae live within these cases that they produce. The cases are usually species specific, so you can identify the species of moth by the larval case. And the larva lives within this case and feeds on various leaves and plant structures as it develops, and then the adults emerge. The adults are sometimes metallic looking. A lot of them are sort of gray and indistinct and look like this. But you normally mostly encounter these by observing the case-bearing larvae in some instance. The blastobacids are another group of Glycioids that are pretty indistinct. They're just little brown moths. They don't really look like much. They are quite diverse in different regions, and so it's something that you potentially could encounter. Really, the distinctive character for this group is the notch and the antenna. You can see just this diagram down here. You can see a little bit in the pictures up here where the antenna is a little bit notched there. Under the microscope, it's a lot more distinctive. And then the Gillikids themselves, and this is the group that contains Tuta absoluta. And as Jim mentioned this morning, a lot of Gillikids you can distinguish by this sort of pointed end of the hind wing. So you have the hind wing that has this little apex or point to it, and that's really distinctive of a lot of different Gillikiids. So you can identify if it's a Gillikiid if it has this hind wing character in a lot of cases. The causids and the Causoidea is another group, another superfamily of moths that contain, hang on one second here, there we go. They contain several families, in this case 7 families. Some of these are distinctive, and you have the causids are wood borers. This is the big moth at the top up here. And then in the middle here, we have this thing that looks like a wasp. And this is actually a moth. We had this example in one of the breakout sessions this morning. This is a Sicid moth, and these are wood borers as well for the most part. And a lot of these moths mimic wasps, and they look very familiar, very much like wasps, and they can be very difficult to separate from a wasp without actually collecting the specimen and looking very closely at it. And so they do a good job of protecting themselves by looking like something that will sting you. There's some other groups within here. This is a Pasendicea archon. This thing at the bottom here is a palm tree pest. That's from originally from South America that's spread to other parts of the world. And so this is a group that contains a lot of important pest species. A lot of the Siciids are important tree pests as well. Zygoniids are another large group of moths. A lot of these are potentially familiar to people living in the tropics, especially their larvae. And so we'll go through 3 of these families real quickly. So the Limacodids, a lot of these are brown or green with these sort of triangular forewings. There's a lot of really brightly colored green species that are really pretty distinctive. And I'll show you the larvae here on the next slide. The Megalopigids are another group within this superfamily. They look very fuzzy. You can see where there's lots of scales on the thorax and legs and abdomen and that. So they look like they're just a very sort of little fuzzy moth. And then the Zygoniids, we had this on the breakout session this morning too. A lot of these are brightly colored, like the one here on the right that occur in the Palearctic. But there's some other things like the one here on the left that looks sort of like a beetle. And we saw some examples of those this morning where you have these, some beetles that look like this and also some arctiids that look the same. So here's an example of some Limacodid larvae. Of course, a lot of these are stinging, so you have to be careful handling anything like this. But these are very diverse in the tropics. If you go to any sort of tropical rainforest, you're likely to see a lot of different varieties of Limacodid larvae, and they're really a diverse group in the tropics. The Pterophorids are a very distinctive group. And that's why we're really talking about them, because they're easy to identify. And they have these divided forewings and hind wings. You can see that the wings are divided into usually 3 different branches. And that makes them really distinctive. And also when they're resting, they look like this T shape, very distinctive if you find them resting somewhere. Really quite easy to identify. It's not something that would normally show up in a pest survey trap, but it might be something that you would encounter just doing a general Lepidoptera survey. The Bombycoids contain the largest, as in physically largest moths. There's about 5,000 described species, and this includes the Bombycids, the Saturnids, and the Sphingids. These are the giant silk moths and the Sphingid moths, or the hawk moths, or the hummingbird moths, or whatever you refer to them as. 2 of the families, the most common families within this group, are the Sphingids. And so these are the hornworms. Also another common name, you can see the larva up here with the horn on the end, down here in the bottom right as well. They have hummingbird mimics. The one in the middle here is called a hummingbird moth. Usually long, narrow forewings. Some of them have very long proboscis. The longest proboscis ever recorded for a moth is in this Sphingid group. I think it was over a foot. And so a lot of these are really quite large moths and really strong flyers. And you might see them hovering over flowers, feeding just like a hummingbird. The Saturnids contain the species that are the largest moths in terms of size in general. These are the silk moths. They have these big, very distinctive triangular forewings. An interesting fact is that the adults have, in most cases, no mouth parts, and so they can't feed. So once the adults emerge, they mate and lay eggs and die. That's really their only purpose. They're not going around feeding on flowers and not like some moths and other groups. Here's some of the distinctive larvae down here in the bottom. The one in the bottom right is an IO moth. And again, that's a stinging caterpillar. You can see the little stingers there on the caterpillar. So something you want to be careful about handling. Generally, again, this is not a group that you would encounter doing a pest survey, but it's something that you might see, just doing a general moth survey, or just see on the side of a building on a light or something like that, because the moths are so large, they're easy to find. Lasiocampids contain the families with the tent caterpillars. You can see a tent up here in the upper right. And so this is a structure that's made by the larvae that live communally. They can cause a lot of damage to trees. These are some important pests in some cases. The tent caterpillar itself looks like the picture here in the middle, and then the adults look like these here on the bottom right for some of the common tent caterpillars we have here in North America. There are some other closely related species down here on the left and at the bottom of commonly referred to as lapid moths. And these guys, of course, are tent caterpillars. Again, nothing that you would probably normally encounter during a pest survey, but you might see the tents on trees in some areas. And so just something to keep in mind if you encounter those. The geometrids are a group that contain a lot of species, almost 24,000 species of geometrids. They're most commonly known for their larvae up here in the top right, called inchworms, because the way they inch along is because they're missing most of their legs. You can see that they only have 2 pair of legs on the back here, and most moths would have more than that. There's not a lot of pests in this group. There's a few different ones, but nothing very common, and I don't think there's really anything in the tropics or Caribbean region that would be considered a pest. But the adults are quite distinctive. They have these very triangular forewings. A lot of the adults are fairly large compared to some of these other groups that we've been talking about. And so again, it's just something that you might encounter doing a general Lepidoptera survey. And so now we've got to the pest groups. And so I want to talk quickly about the 3 superfamilies that contain the most number of pests. And I don't have the Galichioids included here, the group where Tuta is included. They would probably rank, number 4 down here. There's just not a lot known about a lot of the Galichioids, so they're not commonly included in some of these lists, although a lot of them are certainly pests as well. But the noctuoids have the most number of pests. These numbers are from an old list, so there's definitely more than this, but you can just see the relative number of pest species that we consider in these groups. The noctuoids have, in general, the most number of species, and it has the most number of pests in general. The pyrroloids would have the next most number of pests, and then the torticoids have the sort of third most number of pests. And I'm going to go back real quick to somewhere here that Julieta had a comment. Oh, I think I have the, yeah, I have the gypsy moths coming up here. Sorry, I thought I had it previous to this. So we're going to go sort of in reverse order here. We're going to start with the torticoids, and we're going to work up to the noctoids, which have the most number of pest species that we would be concerned about. So the torticoids includes the tortricids. This is the group that I'm a kind of a specialist in, about 11,500 species. These are called leaf roller moths because their larvae often roll leaves, but they have a variety of other feeding styles as well. You can tell the adults sort of just by looking at them based on their sort of rectangular forewings. You can see the forewings of a lot of these different species are sort of rectangular. We won't talk a whole lot more about that, but this is just a group that is commonly encountered during pest surveys. If you put out a Tuta trap, you're likely to get a tortricid in it just by the moth kind of randomly flying in there. When we had surveyed for Tuta in various places in the country, we would always get some sort of local tortricids in the traps. So it's useful to be able to recognize a tortricid versus a Gilligan. The most useful character, other than just the general look of these and that they have these sort of rectangular wings, and you see they don't have a little apex or a little point on the hind wing like a Gilligan would. But if you also look at the mouth parts, the labial palpi don't come up over the head. You can see they're kind of short and stubby and sort of just stick forward. And that's what this term here, porrect, means. Basically, they're just pointing forward rather than coming up over the head. And so it's useful to be able to distinguish tortricids from Gilligans when doing a survey like that. The pyraloids includes two different families, the crambins and pyralids. This is another group with a scaled proboscis. You can see in this SEM picture down here that the proboscis is scaled like a Gilligan. These moths have tympanal organs or tympanum. These are the ears that we talked about in one of the breakout sessions earlier. There's a different form of each of these in each of the different families. I'm not going to talk about that. That's Jim's specialty. But this group contains many pest species. The wax moth is one you might have heard of before. Pickleworm, this thing here, Diaphania, these sorts of things are very common sometimes in the tropics. And so pyraloids are something that you might encounter quite often as well. You can see a lot of these species have some very triangular wings. And again, they don't have this distinctive point on the hind wing like you would see in a Gilligan. The proboscis is scaled, as I mentioned before, and the labial palpi are, again, more like a tortricid in most cases and not coming up over the head like you would find in a Gilligan. And then we come to the noctoids. This is the largest family or largest superfamily of moths. It contains 6 families currently. It certainly contains the largest number of pest species. You can separate the families by wing venation. It can be somewhat difficult to figure out some of these things, but you can generally tell which family they belong to by the way they look for a lot of these common groups. So the notodontids is one of the larger families. This does not contain any major pest species that I'm aware of. Most of these species have very distinctive larvae. You can see a couple examples here in the middle. The adults look fairly distinctive as well. Again, this is not something that you would normally encounter during a pest survey. The eurybids, however, contain a large number of species, and these are things that you would normally encounter, especially in the tropics or in the Caribbean. A lot of these are larger moths, like this one up here in the top. Some of them have very distinctive wing patterns, like this catocala in the middle. And then it contains a group that used to be its own family, the arctiines or tiger moths. The example down here in the bottom right So these are all tiger moths. It's a very large group, very diverse in the tropics, very diverse in the Caribbean. You can see some examples down here in the bottom left. A lot of the adults are very brightly colored. So you have these really bright red and yellow and white spots. Make them very distinctive. And then a lot of the larvae are very, so we call hairy, or have a lot of CD, like this one in the middle here. So it's a very distinctive group, something you might encounter doing any general LEP survey. And again, these moths are fairly easy to recognize because they are so colorful in a lot of instances. The Lymantriids are it's another group that used to be a separate family. These are now under the Eurybids, and these contain one of the most important pest species, the gypsy moth. You can see here's a male down here, and here's a female up here laying eggs. This is an egg mass below the female. The larvae are very colorful a lot of times and have a lot of CD and things, like the making them look very hairy, like these two examples here. They're called tussock moths as the common name because of the tufts of hairs coming off the larvae. And again, these have some very important pest species like the gypsy moth. And the noctuids then, specifically the noctuids, contains probably the most number of pest species. You can get into some noctuid taxonomy based on the different veins and the forewings and hindwings and all. That's way too complicated for this this session, but a lot of the larvae are commonly called cutworms or loopers, and loopers specifically refer to the subfamily Plusiine subfamily that contains a lot of different pest species like this guy here in the middle. This contains, this family contains Lepidoptera. These are really important pest species. Lepidoptera from Asia, Littoralis, more from the Palearctic. Exigua is really common. Frugiperta is the fall armyworm. It's one of the most important pests in the world. It's now been spread recently throughout pretty much the entire world. Amestor brassicacea is a cabbage pest that's really important. Yeah, and somebody texted here, Exigua might show up in your surveys. Exigua and Frugiperta are two of the most common moths in the world at this point. Especially Exigua is one of the most common things that in this region of the world, and so especially in the Caribbean as well. So these are definitely things that you might encounter. You can see that their wing pattern is not too distinctive, but you can also see that they're pretty easy to tell from something like a Gilligan or from Helicoverpa, which we'll see in the next slide. And so the Plusiines include things like Trichoplusianae, other species of Plusiines like Chrysodexes includens. I don't have a picture of it here, but it's one of the most common species in the New World, and especially in the New World tropics, something you definitely might encounter. Copetarsia is another large group of pests. This is in its own subfamily here. I don't think that these are too common in the Caribbean, but they're definitely common within South America and Mexico and some other regions like that. And then the Heliothines, and this includes Zea, the species here that you're going to practice your dissections on. And then the very important Helicoverpa armigera, also known as the old bullworm, which was fairly recently found in Brazil and has spread throughout pretty much all of South America, and is also present in the Caribbean. And so that's something that you might encounter. It's a very, very important pest species. And so the dissection examples that you'll be doing later tomorrow, and I think on Friday, we'll show you how to dissect Helicoverpa, and that skill can be used in dissecting your Tuta as well as other Helicoverpa that you might encounter, including this species. And we gave a lecture on a workshop on old world bollworm in Trinidad a couple years ago. And so we have other resources on that if you're interested. And I think that is the end of that presentation. So does anybody have any questions? I went through that kind of quickly because I know we're running a little bit short on time, but certainly you'll get copies of this presentation. And the idea is just to look through the different pictures of moths and see what you might recognize, and it will help you in doing surveys to recognize the non-targets versus the targets. And there's some of these things, again, like Lepidoptera exigua and Chrysodexes includens that you might encounter commonly during doing surveys in the Caribbean.
Thank you so much. We do have an activity next, but I have a brief poll that I would like everyone to complete before we go into the activities. And I don't see any more questions. I'm looking in the chat. So I'm going to launch this poll and give you a couple of minutes for that. And then we're going to go into our breakout. So the poll is anonymous. So thank you for filling this out so we can kind of understand where everyone is at.
Amanda, the submit buttons doesn't seem to work.
For the poll, you mean?
Yeah.
Well, I'm not sure. This is my first time actually offering a poll. It looks like it's working for some, so I guess if it's not working for everyone, apologies, and we will just
I think you have to answer all the questions.
Yeah, there are 5 questions. So when you answer all, then you could submit.
That may be. Thank you for that group assistance. And while everyone is finishing the poll, I'd like to encourage everyone to, if you don't have specimens, please get some sort of moth you can just even blacklight collect any sort of moth and try to dissect it, start it in the KOH and dissect it. You can also use heating method, which Julieta will be talking about. We listed a hot plate as one of the things for you to have on hand. Okay, we're going to go to our breakouts next. And, in your breakouts, we have the activity, but I'd also like everyone to talk about, hopefully their plans to actually start sharing their screen in their labs. It's important that when you go to the breakouts that you start, moving towards being able to hopefully walk through the dissection tomorrow. And some of you have already taken photographs. That's awesome. And we actually have a lot of people that have already slide mounted, about 2/3 of the class. And most have worked with KOH, but some have not. And I think, and most have not dissected a moth. So tomorrow is going to be really important. And most have their packages, but those that don't, please try to get some moths so that can help. And Sarah and I can be available to kind of help you. Maybe you can reach out to us individually afterwards if you have some one-on-one questions about how we can help you connect a little bit more with showing your lab setup or being able to walk through the dissection tomorrow. So we're going to end the polling and go to the breakout. We will return from the breakout at exactly 11:45 for our final presentation. And so thanks everyone.